Bismillahirrahmanirrahim and welcome you all to the class of animal breeding and genetics. आज हम बात करेंगे इनब्रेड लाइंस पे। इनब्रेड लाइन क्यों ज़रूरी है? पहले तो इनब्रेड लाइंस है क्या, इसकी ज़रूरत क्या है। इनब्रेड लाइन के साथ एसोसिएटेड कई मसले भी हैं, लॉट ऑफ चैलेंजेस है। तो वो चैलेंजेस क्या आ सकते हैं इनब्रेड लाइंस के साथ, और अगर इतने सारे चैलेंजेस के साथ वाबस्ता है तो फिर इनब्रेड लाइन डेवलप करने की ज़रूरत क्या है। तो एडवांटेजेस, डिसएडवांटेजेस और आइसोजेनिक लाइंस और हाइब्रिड विगर और इनब्रीडिंग डिप्रेशन, ये कुछ की कॉन्सेप्ट है जिस पे आज के लेक्चर में हम बात करेंगे। तो सबसे पहले डेवलपमेंट ऑफ इनब्रेड लाइंस, द वाई क्या है? द प्रॉब्लम ऑफ वेरिएबिलिटी। जेनेटिक्स में हमेशा हमारे पास मसला आता है वेरिएबिलिटी का। वेरिएबिलिटी या वेरिएशन अच्छी भी है इन मेनी सेंस, और कुछ सिचुएशन में हम नहीं चाहते कि वेरिएशन। अब हम इनब्रेड लाइन के पर्सपेक्टिव पे बात करते हैं। क्लिनिकल सिनेरियो, एक सिनेरियो है कि अ सेम ड्रग इन द सेम डोज is given to 10 different dogs of the same species, of the same breed, but each dog has different response that is different from the other, maybe little, maybe more. So the same medicine, the same dose given to the same breed of dogs, 10 different dogs, but response is different. Why? Because of variation, because of genetic variation. Each dog, though it is from the same breed, but its genetic composition is different from the other, so the response is different. It's not only the medicine that affect the animal, it's the host response as well, the host interaction with the medicine. So there is variation. The core problem, this variation is the noise. This variation is the noise that makes it difficult to hear the clear signal of the drug effect. इस genetic variation की वजह से, this is the noise, इस noise की वजह से medicine का effect एक जैसा नहीं है, rather is different, it's variable for different individuals. So the disease process is, the drug effect is different, the disease process is different in the genetic, because the genetic predisposition is different. Because of this genetic variation, the effect of the drug is different. Solution क्या है? What if we could remove this genetic variation? अगर हम इस जेनेटिक वेरिएशन को, इस नॉइस की रिमूव कर लें, तो क्या हमारे पास होगा? इनब्रेड लाइन। सो इनब्रेड लाइन इज द सॉल्यूशन टू रिड्यूस द नॉइस, टू रिमूव द नॉइस, टू रिमूव एंड टू कंट्रोल द वेरिएशन, सो दैट वी हैव यूनिफॉर्मिटी, अ यूनिफॉर्म जेनेटिक बैकग्राउंड ऑफ एनिमल्स हु विल बिलोंग टू द सेम लाइन। So definition क्या है इनब्रेड लाइंस की? A population of animals derived from 20 or more consecutive generation of brother and sister mating, full sib-mating, brother and sister mating for 20 generation or even more. At this specific point, the individual produced will be isogenic. All the individual produced, they will have same genetic makeup. They will be isogenic. And isogenic individual, we say when their genetic makeup, their genetic similarity is over 98%, 98.7%. And this is achieved after 20th successive consecutive generation of full sib-mating. Full sib means full brother and sister. So the isogenic individuals are produced, which is intended, which is the inbred line. So how it is achieved is okay, there are three main points: sib-mating, F20 generation, and isogenic. Sib-mating is the most rapid method to increase homozygosity. The most rapid method to increase homozygosity is the sib-mating, full sibs mating, brother and sister mating. This will result in increase in the homozygosity. When we reach to F20 generation, consecutive mating of full sib for 20 generations, we achieve 98.7% homozygosity at any locus, at all locuses. And this is internationally accepted. We say it now, the animals, they are isogenic, they are homozygous, and all the locus, all the alleles, they are homozygous when they achieve 98.7% of homozygosity. Isogenic, all members of the line are genetically identical to one another, clones. Now the animals, they are isogenic, they are genetically identical, like the identical clones, like identical twins. Twins are, they're genetically the same, they're phenotypically the same. Now, at this point, all the individuals, they will be isogenic, they will be similar, exactly the same features, exactly the same color, exactly the same weight, the same height, the same phenotype, and exactly the same genotype. Now the how, the process and its genetic consequences. Kaise? How can we achieve it and the genetic consequences? The journey to homozygosity, homozygosity की करियर सफर कैसे हम तय करेंगे? सबसे पहले हमारे पास होता है founder population, then F1 generation, then successive and consecutive crossing up to F10 generation, and continued for F20 generation. The founder generation is genetically diverse, and they are outbred population. The founder population, they are genetically diverse, and they are outbred. They are from different breeds or from different lines, so their genetic makeup is different. Followed by F1 generation. In F1 generation, there is mating of brother and sister, full brother and full sister mating. And when this is continued till F10 generation, we achieve 88% homozygosity. And if the same mating of full sibs is continued for 20th generation till 20th generation, we will achieve 97.8% homozygosity. And this is the point where when we say that all the individuals, they are isogenic, they are homozygous, and they are the line breeding, the aim is achieved. How this genetic shift occurs from heterozygosity to homozygosity? In heterozygous form, we know it is the alleles are in heterozygous form like capital A and small a. Two different alleles for a gene, and this is common in outbred population. If the population is from two different breeds, so most probably they are heterozygous, or their F1 may be to a larger extent that will be heterozygous. Like AA, homozygous capital AA, if we cross that with small AA, so the F1 generation will be capital A and small a. Homozygous is small, homozygous recessive or homozygous dominant, two identical alleles, and this is the goal of inbreeding, to achieve the homozygous allele. How can we achieve this? Through consecutive mating of full sibs for consecutive generation or up to 20 generations. What happens? There is chances of inheriting some allele, same allele. Chances of inheriting same allele will be high. The same allele will be inculcated, will be disseminated in the offspring, and alleles are fixed after, say it after 10th or 20th generation, alleles are fixed and across entire genome. All alleles, all genes are fixed across the entire genome. So they are identical. What are the challenges? Inbreeding depression. The biggest challenge of inbreeding, of line breeding, is the inbreeding depression. When we go for inbreeding, that is mating of closely related individuals, in this case full sib-mating, this results into inbreeding depression. In inbreeding depression results into decrease in fertility, decrease in performance, decrease in growth, decrease in weight gain, decrease in litter size, and blah blah blah, many characters. This is because in inbreeding, in line breeding, there is a chance that homozygous recessive alleles, they become exposed, they are unmasked, and many of them are deleterious, some are inferior, some results into some syndromes. So inbreeding in most of the cases, in particular situation, it is not preferred. But then why we take this challenge? If this carries so many troubles, so many problems, then why do we go through all these troubles? Because inbred lines it's a powerful tool. It's a powerful tool used in many situations, in many instances. And what are those situations? Drug modeling. In drug modeling, like we have a famous example of onco mouse, then NOD, non-obese diabetic mouse. These are two lines on which specifically designed for drug modeling, and on these specific lines, experiments are being conducted trying different drug models. Then we have drug and vaccine testing. In inbred lines, we have uniform. All the animals are uniform, or they are identical, they are genetically the same. So we get the uniform response. You see the question? We get different response for different dogs, though we give the same medicine, the same dose, the same breed of dogs, but different response. But if we have inbred lines, so all the animals are same. Here they are different. There's variation in the genetic makeup. Here the genetic makeup is the same. They are the clones. They are isogenic. So we have reduced the noise. We have eliminated the noise. We have eliminated the variation. Now the response is uniform. Any medicine we gave to even 10 or 20 individuals, they are genetically the same, so the response is the same. And this can give us accurate assessment of the drug efficacy, the toxicity, and the dosage. Drug जब बनते हैं, it goes through many stages. So सबसे पहले सिमुलेशन स्टडीज होती है, कंप्यूटर बेस्ड स्टडीज, फिर कोई उससे हम कुछ कंपाउंड्स हमारे पास हम कह सकते हैं थ्रू डिफरेंट एनालिसिस की कंपाउंड्स अच्छे हैं, फिर उन कंपाउंड्स को हम टेस्ट करते हैं लैब में, हम सेल कल्चर जाके हम एक्सपेरिमेंट करते हैं, फिर सेल कल्चर के बाद हम उसको एक्सपेरिमेंटल एनिमल पे ट्राई करते हैं. एक्सपेरिमेंटल एनिमल्स आर दीज लाइंस, इनब्रेड लाइंस. एक्सपेरिमेंटल एनिमल के बाद जाके हम स्मॉल पॉपुलेशन ऑफ ह्यूमन बीइंग पे जाके वो टेस्ट होते हैं या एनिमल्स पे. उसके बाद फिर एनिमल का साइज या ह्यूमन का साइज हम थोड़ा बढ़ा लेते हैं. अगर ये सारी चीज़ें एफिकेसी और टॉक्सिसिटी ये सारी चीज़ें नॉर्मल में हो तो ठीक है, नहीं नॉर्मल में तो हम उसको नॉर्मल पे ले आते हैं. उसके बाद इन अ बिगर कोहॉर्ट ऑफ इंडिविजुअल्स द मेडिसिन और द ड्रग और द वैक्सीन इज बीइंग टेस्टेड. अगर वहाँ पे बिगर कोहॉर्ट में इसका रिस्पांस अच्छा हो, इसकी एफिकेसी ठीक हो और इसकी टॉक्सिसिटी नहीं आ रही हो, रीनल टेस्ट और लीवर फंक्शन टेस्ट ये सारे ठीक आ रहे हो, फिर हम इसको रीजनल वहाँ पे जाके हम देख लेते हैं कि वहाँ पे इसका रिस्पांस कैसा है. So the story begins from these inbred lines, from the lab animals and from these inbred lines. Then the other advantage is transplantation study. Syngentic transplant, tissue transfer without immune response. If we transfer any tissue from one animal to the other, from one human being to the other, there is immune response towards that tissue, or towards that organ, or towards the cells. Every individual is unique. The immune system is unique. Anything from foreign, the body produces response to it. The immune respond to it. But if the animals are genetically the same, they are identical, so each animal is, in analogy, it's like the same like the other. So if we remove some tissues from this animal to this animal, there will be no response, no immune response in this animal, because the body thinks it's its own cells, its own tissue. They are the same, genetically the same, phenotypically the same. So this is a good model for transplantation studies, for organs transplant, for tissues transplant. And then comes genetic mapping. These inbred lines are also useful in being used in genetic mapping, like genetic knockdown. We knock down some genes. For example, we knock down the gene of cancer, the gene of some other disease, gene of sleep, gene of obesity, gene of whatever. So we knock out that gene, and then we see the animals, how they perform, how they grow, how they function, how is their life, how is their body function, how is the performance. And the transgenic animals, knock down the transgenic animals and function of single gene. So we can test these animals, we can do experiments on these animals at a single gene level, as well as it is either a fragment of a single gene or a complete gene. So we can knock down and we can induce and we can transplant using this inbred line. Now comes the veterinarian critical role. Veterinarian का being a student of animal breeding and genetics, आपका क्या role है? Colony health management and biosecurity. As these inbred lines are genetically uniform, the susceptibility is uniform. इनकी strengths भी uniform है और इनकी weaknesses भी uniform है. So if there is a one pathogen that is devastating, that can eliminate the entire population. If there is heterozygous, animals are heterozygous, each individual response is different. Some are maybe prone to the disease, some may be resistant, some have hybrid vigor, good health. But in this case, is the strengths are the same, the weaknesses are the same. अगर कोई एक पैथोजन आया जो devastating है इस लाइन के लिए, वो सिर्फ एक एनिमल को किल नहीं करेगा, rather it will eliminate the entire flock, the entire animals. So we need to implement and maintain strict SPF. And second is managing inbreeding depression. We need to recognize and mitigate line-specific health issues. If there are some health issues for some specific lines, we need to understand that and we need to mitigate it at right time. Like hydrocephalus in C57BL10 mice. So it's a line of mice in which this hydrocephalus is a common phenomenon. So being a veterinarian, being an expert of animal breeding and genetics, you need to fix, you need to know, you need to fix the situation of inbreeding depression. Then comes ethical oversight and advocacy. In every institute, in every research organization or teaching organization, there is IAEC, Institutional Animal Ethical Committee, which looks for animal ethics. Though you can use animals for research purpose, but you should also take care of the ethics and the welfare of the animals. So being an animal veterinarian, being expert, you need to advocate for animal ethics and animal welfare as well. Thank you.